Okay, I wanted to go through some of the techniques here just a little bit that you might not have heard of quite as much. So, yeah, that's when I was looking to see if there was supplementary information. I don't see it, or it's not called that. So, this is the way a materials and methods section usually starts. Normally, if it's something to do with a virus or a vaccine, your first section is going to be cells or cells and viruses. And so here they're just listing all the different types of cell that they're going to use and exactly what kind of food they're going to feed them, which is mostly going to be some version of this thing. Delbecco's modified Eagle's medium, DMEM. That's very common. And we do put in fetal bovine serum, which is serum like blood minus cells from baby cows. Yeah. And they put this in because it has a lot of good growth hormones that really help the cells you're trying to grow to stay alive and keep growing. But yeah, it's kind of gruesome at the same time. Let's see, plasmids. This is how they're going to make their different MERS coronaviruses. And so they get a plasmid with the nucleoprotein in it, and eventually they're going to build up a rabies virus plasmid that expresses either the nucleoprotein gene or the spike gene of MERS coronavirus. I think this paper is mostly about the nucleoprotein gene. All right, so let's go down here, past the viruses. Yeah, MERS, and oh, there we go. Here's the measles vaccine with the MERS S, and they also make a measles vaccine with the MERS N, and a measles vaccine with just green fluorescent protein. All of which they can use in different ways to kind of track it. So very similar to the last paper. All these people are sort of swimming in the same pool of ideas, and yeah, it all makes sense. But this is why I wanted to do this one, because it follows nicely from what we were just talking about. Okay, Western blot. What's that? You might have heard of it already. This is a thing where you look for proteins. And you're going to run a protein gel just like normal. You're just going to push all the proteins down, sort them out by size. But then at the end, to find your very most special protein, you're going to put an antibody on against, for example, the coronavirus N. And that'll show you which of these proteins, because there'll be thousands on that gel, is actually the one that you want. And then you can also check, you can compare it to some proteins of known size. You normally run what they call a ladder of those. And that'll show you that, yeah, you're the right size, and you're binding the antibody as if it was the nucleoprotein. And with those two pieces of information, we can say, yeah, it's probably the nucleoprotein. Skip animal experiments, although there are good animal experiments here that we're going to have to look at. Actually, no, we will mention, okay. So, IFNAR knockout mice is what this is. So, this is interferon type A receptor, and minus minus means they are missing both copies on the chromosome from mom and the chromosome from dad. So, these things are totally deficient for receiving interferon receptor. And this is usually associated with really severely immunocompromised mice. These things will die at the drop of a hat, basically. And I believe that's what they had to do to get a mouse model where you got some disease from MERS coronavirus. So they could do the challenge, basically. Okay, neutralisation assay, that's a virus neutralization assay. That's the same as the stuff we've already covered, so we don't have to do that. These are the two I wanted to mention because these are kind of specialist immunology assays, but they both look for T cell function. And so at the top, we've got here an ELISpot assay, and at the bottom, we've got an intracellular cytokine staining, which they call ICCS. They're both looking for the same thing. These are both ways to see whether T cells have started to work. And so the way you do this is that you take white blood cells, some of which will be T cells, and that's fine. You can sort them out, you can put them in a dish, and so there, you've got a little dish of all the T cells from, a milliliter of mouse blood, something like that. Then, in order to see what these T cells react to, you can feed them little peptides, little pieces that come from the virus. And so you can feed them little pieces of peptides from the spike protein or from the nucleoprotein, or just a sort of a junk control peptide that they probably shouldn't bind. And if the T cells react to it, what they'll do is they'll start producing interferon gamma. That is their message for, I got a hit, eureka, I found it, whatever. It's their little sort of, shout for joy when they've found something they can recognize. And so there will be some portion of the T cells in this population that react to each different part of the nucleoprotein or the spike protein. And this is how you find them. You just keep, you say, okay, here's a little piece of spike. What do you think of that, T cells? And some of them will say, yay, interferon gamma. And the rest will just sit there. So in the ELISpot assay, they're actually going to make a little purple spot at the bottom of a well. And that's going to be due to the cells reacting, showing that they are awake. In the intracellular cytokine staining, which is a little more difficult and more old-fashioned version of the technique, you're still going to feed these little T cells a peptide and see if they like it. But then you put in a thing called Brefeldin A, which is on here somewhere. And what Brefeldin A does is, there we go, Brefeldin A actually blocks transport from the ER to the Golgi. So it's going to stop it right there at the transport stage. And so what that means is that the cell is going to recognize a peptide, and then it's going to try to make interferon. And instead of just spitting the interferon out, into the atmosphere, where you would then have to try and track it down and figure out where it came from, what you do is you just jam the transport system right here. And so the cell will fill up its endoplasmic reticulum with interferon gamma, little tiny proteins. And then you can go in and stain for those with another antibody, and that's how you can find whether you have a T cell that has successfully reacted. And so that is what we're doing here. In one of these assays, you actually have to give them dendritic cells, here we go, which are going to present little pieces of the protein to the T cells. You can also do this with just the little tiny bits of protein themselves, just like a little peptide all on its own, and that will also work. There's our Brefeldin A. There we go. So that's these two assays in super brief form, and there's a nice Wikipedia page on each one, and that's probably a good place to start for figuring out how to tackle those particular parts of the paper. So thanks very much.